Hello all, welcome to the very first installment of Parlor Paleoanthropology, part of the Wild Scientist podcast series. An introductory podcast to the wonders of the archaeological record, hominin fossils, and, of course, human evolution. I'm your host, Anna Tulley. Joining me today is ISU alum Quinn O'Mara.
Hey everyone.
Before we get to the podcast part of the podcast, I'd like to thank some of our sponsors. This episode is made possible with the support of the Department of Sociology and Anthropology at Illinois State University. A big thank you to Dr. Shelby Putt, my paleoanthropology professor, for her guidance and feedback throughout the podcast making process. Now, on to the good stuff. Today's episode is about tapirs.
Wait, like the little pig-looking animals?
Exactly. As it turns out, tapirs can tell us a lot about Paranthropus boisei.
Huh. Who would have thought?
Right? So, let me set the scene for you. Paleoanthropologists have been studying the diets of hominins for years. What did human ancestors eat to survive? Although the fossil record has provided us with a pretty good idea, the conclusions aren't always obvious. Larissa DeSantis believes tapirs are the key to the truth about one hominin's diet. DeSantis is a paleoanthropologist and associate professor in the Department of Biological Sciences and Department of Earth and Environmental Sciences at Vanderbilt University in Nashville. She holds degrees from University of California, Berkeley, Yale, and University of Florida. DeSantis has published more than 50 papers and book chapters, and her work has been featured on CuriosityStream, National Geographic Wild, the Discovery Channel, radio shows, and global news coverage.
Yeah, I trust her.
Yeah, me too. In June of 2020, DeSantis and her team published a study in the journal Scientific Reports,
clarifying relationships between cranial form and function in tapirs with implications for the dietary ecology of early hominins.
Right. In this paper, she explores the correlation between sagittal crest morphology and diet, specifically durophagy, in several extant and extinct tapir species.
I'm gonna need some definitions here.
Okay, let me break it down for you. The sagittal crest is a bony ridge that runs along the top of the skull, providing extra surface area for the attachment of the jaw muscles. Historically, a pronounced sagittal crest indicates incredibly strong jaws. Durophagy is when organisms eat hard foods, such as crabs or hard-shelled nuts. And extant is just the opposite of extinct.
Okay, that's all easy enough. But what does this have to do with paleoanthropology?
Well, the researchers wanted to know if tapirs' large sagittal crests are required for eating hard food, or if they had other functions that we didn't know of. If they could crack the case with tapirs, which have similar sagittal crests to certain hominins, then this knowledge can be applied to our human ancestors too. You see, Paranthropus boisei is often thought to have had a hard food diet, filled with nuts and seeds. In fact, they nicknamed him the Nutcracker Man. But a dental microwear study conducted in 2008 by Peter Unger suggests Paranthropus boisei ate tough, fibrous foods rather than hard, crunchy ones. Therefore, a pronounced sagittal crest doesn't necessarily equate to durophagy. DeSantis' study expanded on this idea.
Excuse me. Dental micro what?
Basically, when you chew, your food leaves microscopic marks on your teeth. Dental microwear texture analysis, which is a mouthful, so we'll abbreviate it to DMTA, analyzes these teeny tiny indentations to determine mastication behaviors and dietary features. DeSantis and her team determined that despite variable sagittal crest morphology in tapir specimens, they couldn't find an apparent relationship between DMTA attributes and sagittal crest development. This idea can then be applied to Paranthropus boisei.
Okay, I follow you. What else did they find?
Well, DMTA is incredibly useful on its own, but DeSantis and her colleagues were practically unstoppable when they used it in combination with finite element analysis. And before you even ask, let me explain. Finite element analysis, or FEA for short, is the digital simulation of a physical behavior through mathematics. They recorded the stress distribution on several specimens of tapir during unilateral biting at the second and fourth premolar and third molar. Their findings suggest the presence of a sagittal crest can affect the energy efficiency and strength of the cranium in tapirs and carnivorans. Increased masticatory muscle volume, which needs a place to attach, may help process greater amounts of vegetation with less muscle fatigue. This would explain Paranthropus boisei's morphology. By imagining modern tapirs with known diets and a variety of tapir fossils from throughout the southeastern United States, including the Gray fossil site in East Tennessee, they found that the larger crests were absent in tapir species that consumed the hardest foods, but are present and even quite pronounced in species that chewed for long periods of time on soft but tough foods. Together, FEA, DMTA, and ecological data suggest that the presence of moderate to large sagittal crests in extant tapirs does not correlate with hard object feeding. Rather, it correlates with processing of tough or less nutritious vegetative matter. This means a more folivorous diet.
Okay, this all makes sense. But can we go back to how tapirs can be compared to Paranthropus boisei? I'm having trouble seeing the resemblance.
I don't blame you. On the surface, tapirs and robust Australopithecines seem to fall on opposite ends of the mammalian spectrum. However, tapirs are actually the perfect candidate for proxy analysis because of their herbivorous diet and variable sagittal crest development within the genus. Unlike the sexual dimorphic differences in previously studied organisms, such as great apes, sagittal crest morphology in Tapirs is not correlated with size, with Tapirs terrestris being intermediate in size, but with the tallest crest. Such variance adds depth to the data, allowing it to be applied to other large mammals, including Paranthropus boisei.
So what does this do for the field of anthropology? I mean, such a broad comparative analysis could definitely be replicated with other species, right?
Absolutely. These researchers were definitely imaginative, and it paid off. DeSantis even said, "Sometimes you have to think completely out of the box and rethink relationships between skull shape and function. Since there are no living primates resembling robust Australopithecines found around today, proxy studies are the key to understanding ancestral hominid diets. Looking at a spectrum of mammals may help us understand other early hominid behaviors rooted in morphology." The bottom line is, we have to reconsider the relationship between form and function. Without such an innovative re-examination, how would we have known that Paranthropus boisei was likely munching on tough grasses instead of cracking seeds? That's pretty cool, right?
Yeah, I'd say so. I guess this means we should say goodbye to the Nutcracker Man name, huh?
Yeah, I think, Gritty Grass Guy has a much better ring to it anyways. And just like that, we've reached the end of the very first episode of Parlor Paleoanthropology. I'd like to extend a special thanks to Dr. Larissa DeSantis and her incredible team of researchers, all of whom made today's episode possible. And thank you to Quinn O'Mara, my lovely partner in crime, for lending his voice and thoughts to this project. Finally, thank you to all the listeners for taking the time to learn something new today. See you next time!